Right, now I'd like to introduce Professor Hawkins. I'm thrilled that he's come, and I met him once on Lundy whilst I was trying to swim with seals, and he was taking some super photographs. So I don't know if he remembers that, but it was a very important moment for me because I got an introduction to true expertise about life on the shore there. Early introduction. So welcome to Professor Hawkins. He's a very eminent rocky shore scholar, well known to most students of marine biology for Intertidal Ecology, co-authored with David Raffaelli. I have acquired a copy and I am fascinated by it. I hope some of you will do the same. Endearingly, he says on his University of Southampton website that he likes to be on the shore counting things, which will commend him to all of us, I think. But even more important for us is the fact that he comes from Braunton, a wonderful place, centre of the universe, as you know. He's a good friend of Pip and Pete Jollens. Pip's with us today. They've been great supporters of Coastwise. They met Steve in Liverpool University and have often mentioned him since, obviously with great fondness and great memories. He embarked on an illustrious academic career after his first degree and doctorate, including professorships at Plymouth and Bangor, as well as his first and current, emeritus chair at Southampton. Intriguingly, I hope he'll tell us more about this, incorporating periods as director of the MBA in Plymouth, where he's now a vice president. His ongoing research interest and extensive list of publications reveals that he has never lost his fascination for studies of barnacles, top shells, limpets, and the like. He was a major contributor to the MBA's Marklim climate change project 15 or so years ago, which was really Coastwise's first introduction to Marklim's work and to his work with it when we first started, something I'd like to know much more about, and I hope he'll tell us some of the outcomes. This demonstrated the importance of systematic long-term studies of marine invertebrates and helped awaken our interest in them, studies we look forward to hearing more about. Well, that's quite a formal introduction from me, but I think we're going to hear lots more about his work, and I can't commend it to us enough. And I hope out of it will come out as really enthusiastic onshore surveyors, and that we'll have a chance to be so very soon. Can I hand over to Professor Hawkins now, please?
Okay, thanks so much, Paula. It's great to be able to do this, and it's a shame I couldn't do it back in November, but I was moving house the day before and was in temporary accommodation in Cornwall doing some shore surveys in lockdown. I actually took my Devon sticker off the back of the car so I could blend into the background down there as somebody from outside, but I was actually working and homeless, so I had two good reasons to defy lockdown in Cornwall. Okay, well, what I'm going to talk about today, I'm going to talk for about 40 minutes or so. I'm going to give a very brief introduction about why I became interested in marine biology and actually the role of North Devon in shaping that interest. I'm going to talk about rocky shores as an experimental system for general ecological concepts. And then pause for coffee and a few questions. And then I'm going to finish off talking about rocky shores as climate indicators. And then if I have time, link the two by a brief consideration of the aftermath of the Torrey Canyon oil spill, which sort of brings the two strands together. So here we go. So I was actually born in Weymouth in Dorset, and from a long family with maritime connections, my first introduction to marine biology was on Weymouth Beach, fishing with my dad and collecting slipper limpets for bait. And when I was recently doing some field work in Weymouth, I came across this engraving on the pub wall, and it shows one of my ancestors, Richard Hawkins, who was an excise man being whipped to death by smugglers in the Black Dog pub, which is Arrowed. It's still a pretty dodgy pub. It must have been pretty dodgy in the 18th century. And then we moved to North Malton, where I got interested in freshwater biology. And then when I was 12, we moved to Braunton. And on the top right, you can see me grasping a plywood board, which was given to me in 1968, my first Christmas in Braunton. And I took it out for a surf this year when lockdown was briefly ended. And of course, as a kid in Braunton, I spent a lot of time fishing in the Torrens estuary. I spent a lot of time mucking around Braunton Great Marsh, well, Great Braunton, slightly smaller marsh these days, and quite a bit of time at Salt and Sands, both fishing and surfing, and also at Down End fishing and Ilfracombe Pier. But at school, I was being groomed to go and do medicine. And it wasn't really until I went on a field course on Lundy with my people from Barnstaple Grammar School. It was then just part comprehensive, that I got more interested in the natural world. And it was really that field course that that influenced me not to do medicine, but to go and do a more general biological sciences degree. In fact, we had excellent biology teachers at Barnstaple Grammar School, Park School, and one of them, Mr. Wills, said to me, "You're far too good a biologist to waste it on medicine." And that and the field course got me interested. And in fact, the first bit of seashore ecology I did, and this is my, on the left-hand side is my notebook, which I found when I was tidying the house up when we moved, and actually shows zonation on Lundy Landing Beach, as drawn by a 16-year-old on graph paper. And it was generally pretty well. I spelt most of the names right, didn't underline them, hadn't been to university yet. But I actually read the discussion and it was very interesting. It was very much dominated by physical explanations of the causes of these zonation patterns. And as we'll subsequently come on, there's a lot more to it than that. So I didn't know at the time I'd end up working on quite a bit over the next few years. And really, it was Lundy that was instrumental in making me decide to be a biologist in general. And I applied to Liverpool because I could do 2 years of general biology, keep my options open, and then if I wanted to, I could then go to the Isle of Man for my honours year. Rest in peace, Liverpool University very sadly closed the lab in 2016 after 140 years. And it was on field courses from Liverpool with George Russell, which Pip demonstrated to me on. I got interested in limpet grazing, particularly looking at the seaweeds growing on limpet shells compared to those in the surrounding area. And George Russell really got me interested in plant-animal interactions. And Richard Hartnell gave some good lectures on rocky shores, and I decided to do a rocky shore project. So I've had a long and intense affair with limpets over many, many years. And it was great that in 2015, we actually had my retirement conference in Liverpool. And this is my favourite pub in Liverpool with a sloping floor. As a short ass sort of bloke, it's quite good because you can talk to sort of taller people downstream from you, from where I took this photograph. And of course, it's sloping because in the old days, they used to be able to get all the spit and vomit out of the door with a brush, and also the sawdust. And in 1975, that's probably the last time, maybe the last time I saw Pete and Pip. That's what I was like as a student, but it was great 40 years afterwards to have a conference in Liverpool and meet a lot of my former PhD students, many of whom are from European countries. I've benefited from more than 20 EU students, which sadly won't be available to British academics in the future, but we managed to get together and talk about stuff, basically biodiversity and ecosystems in Liverpool. And I've already had a plug for one of the books. The Rocky Shore Guide is actually the most useful, I think, to amateur people, and this was produced by the Marine Conservation Society as an early attempt to get some citizen science going in the 1980s and 1990s. You can get it on Amazon, I think, and that's out of print, and Intertidal Ecology is out of print, but you can also get that secondhand. There's a lot of students that bought it and then have sold it subsequently, I think. And a more recent thing which came from the Liverpool conference has come out, and if you're really interested, you can perhaps get that via a library to read. So I'm going to talk about, in the first part of the talk, why rocky shores are so good to enable an experimental approach. I'm going to touch on environmental gradients. I'm going to touch on a few key concepts and classic experiments. I'm going to use illustrations work done by myself and my team of hardworking PhD students and postdocs over the last 40+ years. Firth, a lecturer at Plymouth University, demonstrating zonation on an exposed rocky shore, typical of the North Devon coast, where you got a midshore dominated by mussels, and the higher shore bit is dominated by barnacles, giving way to lichens and things at the top. So zonation's a pretty standard feature of rocky shores. And if you go from exposed headlands, they're dominated by barnacles. Again, you can see some evidence of zonation there with seaweeds at the bottom, barnacles in the middle. And if you go to more sheltered shores, this is on the Isle of Man, they're dominated by seaweeds. So I'm going to argue that rocky shores are the equivalent of the lab rat, the fruit fly, or even C. elegans, the geneticist worm of choice as model organisms. And why are they so good to study? Well, they're two-dimensional. You can easily see them. You can do non-destructive sampling. It's not like sampling in mud where you have to sieve it and you can't see what you're going to get till you sieve it. That's fun in its own way, but it's a bit like a lucky dip. The other thing is the organisms, both plants and animals, compete for an easily measured resource, space to live, either to enable them to grow a canopy if they're large brown seaweeds, or space to forage if they're limpets, or space to filter feed if they're barnacles. Most of the species have short lifespans, except for Ascophyllum that might genetically live for over 100 years, and individual clumps might live for 50 years, but most of the species have quite short lifespans. They're manageable sizes, and a lot of the key players don't move around a lot, which makes them easy to track or stick labels on. They're also extremely easy to experimentally manipulate because of the steep environmental gradients. You can move things around from exposed headlands to sheltered shores, see how they survive. You can transplant things up and down the shore. And there's few taxonomic problems. There's a few. The more knowledgeable people will probably spot several name changes as the ages of my slides change during the course of the talk, and we can perhaps ponder on that over a cup of coffee and questions. But above all, experimentation's easy. You can transplant things, you can exclude them, you can remove them, you can thin them, you can do all sorts of things with them, and you can set up adjacent controls to them which are untouched. And the other thing is you get very rapid response times. I was able to remove it during my PhD kelp canopy, and within 3 years I got kelp canopy back again. Had I done the same experiment on an oak woodland, I'd sort of have to wait about 500 or 600 years and put the original experimental design in a time capsule. So you get very rapid response times. So lab rats have made a real big contribution to ecological theory. Concepts like keystone species, competitive exclusion, succession and disturbance, supply side ecology, how recruits can influence population dynamics, positive interactions, how species can be kind to each other, particularly by providing shelter from harsh conditions, and also bottom up and top down effects, which I'm going to dwell on later in the talk. So all that led to this book with David Raffaelli, which came out originally in '96. And a lot of the rest of the talk depends on this. So if there's any copyright police out there, I'm plagiarizing myself. So if we look at the vertical gradient from sea to land, it's a very sharp gradient of immersion from basically fully marine conditions to fully terrestrial conditions. And most of the things that we see on the seashore are of marine evolutionary affinity. You get very few insects, a couple of spiders, a couple of pseudoscorpions, lichens may be of terrestrial origin, but most of the species, all the algae, most of the invertebrates that you see on rocky shores are marine and evolutionary affinity. They've got first cousins and second cousins below the low water mark. So for them, it's a unidirectional stress gradient as you go up the shore. The reverse applies for any terrestrial species. For instance, in salt marshes, the stress gradient goes down the other way because all the salt marsh plants are basically higher plants and they're colonizing a much more stressful saline environment for them. So most species have marine origins, and therefore increased shore level it leads to increased stress. And these could be physical stresses, mainly to do with heat and light and desiccation. They can be chemical stresses, particularly in rock pools due to variable salinity. They can be biological stresses, less time to photosynthesize, less time for algae to take up nutrients because they don't have roots. And they can also be constraints on animals, less time for feeding and respiration if an animal's got gills. It's harder work walking in air if you're a crab than swimming or crawling in water. And above all, you've got much greater variability as you go higher up the shore. All these factors fluctuate, and this can prevent physiological acclimation during tide out periods, and a need to very sharply adjust from when the tide's in to when the tide's out. So we're going from highly stable marine environment. And remember, life evolved in the sea to something approaching terrestrial over about a few hundred meters at most, often 5 or 10 meters on a very steep sheltered shore. And this is the equivalent of going up 3,000 meters of altitude on a mountain in terms of the changes in pattern along that environmental gradient. So those are the physical and chemical factors, those are the biological factors. And that's the key thing, I think, unpredictability. And on this sharp environmental gradient, just as going up a mountain, you get these very sharp transitions, you get clear zonation. This is one of my favourite shores in Swanage, Peveril Point, where you get a very sharp transition from areas dominated by seaweeds to areas dominated by limpets and a few barnacles. There's not much larval settlement there, so you don't get so many barnacles. Now, until the 1950s, all the explanations of zonation and all the ones I wrote in my notebook on Lundy in 1972 as a 16-year-old student were all about the direct response to the physical environment, physiological tolerance along this gradient. By 1972, due to the work of Joe Connell and many other people, it began to be realised that maybe upper limits were set by physical factors, but lower limits of a species on the seashore was mainly set by biological interactions, particularly competition, predation. But since the 1970s, there's been quite a lot of emerging evidence that that is not quite true. And some of that was work that I've done that I'm going to touch on later. So this is a sheltered rocky shore on Anglesey, and you've got some clearly zoned species, Pelvicia, Fucaspiralis, and Ascophyllum nodosum, various types of rack, channel rack, spiral rack, and egg rack. And along the shore gradient, as you go from unfavourable to favourable conditions, you get much less biodiversity, you get much less productivity, you get much more occupation of surface, much more competition for space. Habitat structure, which is biologically generated, goes from very simple to very complex. And generally, the species richness goes from low to high. And the great thing about rocky shores is you can see the pattern, and then you can do experiments, testing hypotheses to understand the processes which are determining those patterns. And these are some experiments that a Portuguese student of mine did in Portugal and in Swanage when she was working in Southampton, just to look at generality of process on the process. And I'll come back to those in a minute. So I'm going to talk about, and in fact that last slide is evidence that upper limits can be set by biological factors, in this case grazing. Because if you look both on the left and the right, you can see that Deanna had managed to induce extension of seaweeds higher up the shore. And she did this by building little fences on the beach and excluding the grazing limpets. So it wasn't physical stress that was stopping these red algae going higher up the shore. It was primarily grazing by limpets that was stopping them going up. Ultimately, physical stress would have stopped them going all the way up the shore, but the immediate boundary was set by limpet grazing. So an example of upper limits for low shore species being set by biological interactions. I would still say that for most upper shore species, physical factors directly set their upper limits, but for mid-shore and low shore species, it can be competition on sheltered shores, and it can be competition. But the ultimate factor is the environmental gradient as you go from fully marine to fully terrestrial conditions. So now I'm going to talk about grazing, and obviously this involves limpets, but also top shells and other grazers. And I'm going to talk about grazing mechanisms of biofilms, I'm going to talk about classic early work on the Isle of Man, and I'm going to talk about work on grazer exclusions and scaling this up with European scale experiments. So I'm going to wave my Manx flag. So if you go from an exposed headland to a sheltered rocky shore, you go from shores dominated primarily by limpets and barnacles to shores dominated by seaweeds. And there's clearly some kind of dynamic balance going on between grazers like limpets and macroalgae. The environmental gradient horizontally, though, is not so straightforward as the vertical one, because there's both benefits and stresses as you go along the gradient from shelter to exposure. And many species have their optimum conditions halfway along, or 2/3 of the way along, or 1/3 of the way along on this exposure gradient. So this isn't a unidirectional gradient. But generally, as you go to more exposed conditions, animals win. There's probably greater supply of larvae there. There's more food for suspension feeders. There's greater oxygen and nutrients. And as you go to more sheltered conditions, where seaweeds can stay anchored, they don't take roots, they don't have roots, where seaweeds can stay anchored, then the seaweeds tend to win. But what determines directly these distribution patterns? And on moderately exposed shores, where the balance can flip between limpets and barnacles and seaweeds, you get quite a lot of fluctuations. This is a shore on the Isle of Man. Sometimes it's dominated by barnacles, sometimes it's dominated by seaweeds, sometimes it gets dominated by barnacles again. And this moderately exposed shore is just at the tipping point between limpets winning and seaweeds winning. So what do limpets do along this gradient? Well, first of all, they go off foraging from their homes. This is a picture taken at Hartland Quay, where there's quite a bit of silt in the water, and the silt has picked up the mucus trails of both top shells and also limpets in the top right-hand corner. So limpets go off and forage. And when they're foraging, they use their really hard teeth, one of the hardest compounds in the natural world, toughened with iron, to actually scrape the rock. Not only do they eat the food, but they eat the plate as well. And they feed on biofilms. They can also feed on macroalgae as well. But a lot of the time, they subsist on a gruel of biofilms that invisibly coat the rock. That's why rocky shores can be very slippery. There's an invisible felt of all sorts of things there. These are teeth marks of various types of grazers. Ocelinus lineatus, previously called Monodonta, now called Phorcus lineatus. Top shell has a brush-like radulae, and the marks that it makes are shown in picture A. Winkles have a rake-like radulae, and those are the marks made by the common winkle, the Trinoliteria B and C. And limpets have a radula like a bulldozer, and different species make different marks. D, E, and F are made by Patella vulgata, and the bottom right is made by Patella aspera, which actually excavates calcareous algae and has a much deeper rasp into the rock substrate. I got these by conning limpets to feed on a plastic Petri dish by rubbing a bit of seaweed on it, and the top three were done by a colleague, Dave Watson, by putting a smoky film on glass slides and conning the snails to graze on them. And this is what they're feeding on. The top right are diatoms, the top left and centre are diatoms, top right are sporelings of green algae probably. The long filaments, which we misidentified in a paper, are cyanobacteria, cyanalgae, as I used to be when I was a student, which coat the rock. And then there's various types of diatoms. These are all viewed under electron microscopy, and these are very important. They're probably the equivalent of grasslands on rocky shores. Not very much biomass, but rapid turnover as they rapidly proliferate. So they sort of live in a film of mucus. And in fact, some limpets even feed along their own trails, and they actually consume pelagic diatoms that cascade out of the water column and stick to the mucus. So they're recouping some of the energy spent on mucus production. So biofilms are very important for primary productivity, particularly that productivity which is available to grazers. But they're also a really important settlement site. That's a barnacle larvae that's 1 day old. That's a barnacle larvae that's 2 days old. That's one that's 3 days old. And that's an adult barnacle. And they actually settle in the film, and they're actually attracted to different types of microbial films. Though they're mainly attracted to settle next to adults of the same species, because these are obligate cross-fertilizing animals, and they need basically to settle within willy length of each other. And these are macroalgal propagules. This is possibly a baby rack plant, a baby fucoid, again seen in SEM. And it's interesting to measure grazing intensity and pressure. And one of my very talented students, Richard Thompson, who now works on plastic pollution, just got an OBE for it and became a fellow of the Royal Society, did his PhD with me on limpets. And he came up with a cunning plan for measuring limpet grazing activity by getting, ironically, plastic screw covers, sticking them on in holes drilled on the shore, filling them with dental wax, and then basically getting impressions of radulae on the rock surface left by the limpets. And if you have a big enough array, you can measure both the intensity and the spread of grazing, and do this in both space and with season. Now, one of my lecturers that would have lectured to Pip and Pete, Norman Jones, did some really interesting work after the Second World War, and he experimentally removed limpets, first from a patch at the top of the shore, which he published in Nature in 1946, and then subsequently from the whole shore, which he buried in the proceedings and transactions of the Liverpool Biological Society, which still had intellectual pretensions in the 1940s and 50s, but not when Pete and I were students in the 1970s. And he showed that in the absence of limpet grazing, and he killed all the limpets on this strip, that seaweeds would proliferate. And it wasn't wave action that was stopping these seaweeds growing on exposed shores. It was actually the direct action of limpet grazers themselves. So he showed that the limpet grazing was preventing proliferation of brown seaweeds, fucoids, racks, and also green seaweeds as well. So grazing rather than physical control was setting these horizontal distribution patterns. Now, it's a very cynical PhD student. I didn't quite believe it. So if in doubt, repeat it yourself. And I removed not a 10-metre wide strip, but a 2-metre wide strip just round the corner. And after 2 months, there was lots of green algae. And after 9 months, being the 1970s, I managed to induce a really nice mohican of fucoids down the shore. And then a few years later, the limpets from both sides came in, munched away on the fucus, and because they'd moved in, meant the grazing either side was less extreme, and there was a further proliferation of algae. So I rather muck that shore up for the next 5 to 10 years. But fortunately, help was at hand, and that was in a compressed air powered drill from scuba cylinders. And I was able to do much more subtle experiments and build, in the top left, little fences on the beach, conveniently labeled treatment with an adjacent control, and was able to show that if I fence limpets out, that very rapidly, within 9 to 10 months, you get this very dense growth of fucoids. And I was able to do various experiments on interactions between limpets and barnacles and fucoids. But limpets basically rule on moderately exposed rocky shores. So that's a mid-successional plot where green algae are giving way to brown algae. So the green algae coming first and the brown algae coming afterwards. And this is a moderate exposure, the moderate exposure on the Isle of Man, which I showed earlier with these fluctuations. And when Norman Jones died, his widow, Joanna Kane, Mrs. Jones, who was also a lecturer that I had, scattered his ashes on the site of the limpet removal strip, and there was a very spooky large recruitment of large brown seaweeds, which maybe the ghost of Norman was doing an experiment. So grazing has a strong effect on both macroalgae and microalgae, and we were able to explore this with some help from the European community throughout Europe, comparing the importance of grazing in the cold north compared to the warm and sunny south. Again, using cages, again, using a very, very complicated experimental design. So managed to get it published in big journals, where we compared different locations. We had to have replicate shores within each country. We did summer and winter initiations because of harsh and less harsh physical conditions for seaweeds. We had to have random dates within summer and winter, and then we had to have spatial replicates within that. It was a very, very complicated experiment. And those are the treatments. We had a fence control where we had limpets partially fenced, but let them come in just to check if there was any effect of the fences, and we had an unfenced control. And we got the same effects on the Isle of Man, and then we were able to compare what was going on throughout Europe. And actually the effects on the Isle of Man in the north were strongest. There's far more fucus there, they grow more quickly, but in every case there was very little fucus in the controls. In the Isle of Man you do get natural escapes of fucus because the fucus do quite well, but nothing like in the exclusion areas where the limpets have been kept out. In the southwest of England it was a bit more variable. In northern Spain, which is a fairly cold and damp region, it was fairly variable. But as we went into Portugal, we didn't get much fucus, and that's because there's very little fucus there to colonize. But the ephemeral algae was still controlled by limpet grazing. And we got different sequences of successions. We got greens sometimes going directly to fucoids in the Isle of Man, or sometimes straight to fucoids. In South Devon, we got greens going to racks, fucoids, maybe crusts, maybe turf. In northern Spain, we got greens going to fucoids, crust and turf. And in Portugal, we got greens going to bonsai fucoids, these small hardy fucoids that only occur in brown algae that occur in southern Europe. So we've got increasing grazing pressure as we go further south in Europe. There's more species of grazers, there's more physical stress. So the probability of a poor baby seaweed escaping limpet grazing or grazing by other grazers such as top shells become much, much less in southern Europe. So to summarize that, grazing controls algae on barnacle-dominated shores throughout Europe. Limpets are a keystone species. You get greater and more predictable effects further north. Physical factors, season, latitude influence colonization rates. And there's quite a lot of variation in early colonizers, which tend to be opportunistic, but the pattern becomes more uniform later on as succession begins to take over. So what about the vertical gradient zonation? Well, this is Diana Boaventura's experiment showed earlier. And what was nice about this was we showed generality of process. The same kind of things go on in Portugal, where it's really hot, as in the Isle of Man. We managed to show that the upper limit of low shore red algae was set by limpet grazing. And there's very much a dynamic balance on this gradient where suddenly the seaweeds win lower down, and actually you have to remove the seaweeds to get limpets to go lower down the shore, and the seaweeds grow so fast they swamp the limpets. But at some point on the gradient, the growth of the seaweeds is slow enough that they can't escape the limpets unless you remove the limpets, and that allows them to sneak up the shore a little bit. So seaweeds are prevented from extending up the shore by grazing. Not a physical factor, desiccation, but actually grazing. But the physical factor is underlying the ultimate factor affecting the performance of the algae and the likelihood that they can escape limpet grazing. So let's go on to other grazing effects, and let's talk about what regulates biofilm. This is also work done by Richard Thompson, which I was a co-author and supervised. And here we were looking at very much the influence of top-down control by grazing versus what's known as bottom-up forcing by light and nutrients. So who wins, physical factors or biological factors, or is it a combination of the both? And how they affect these lawns of diatoms and cyanobacteria. Well, Richard studied the amount of chlorophyll, the amount of green pigment on the shore as an index of how many microalgae were there and cyanobacteria were there, and got this interesting pattern. Actually, the microbial film was depressed in the summer of most years, and its best growth was often over the winter. And if we look at insolation stress, this was stuff from Ronnersway Airport on the Isle of Man in terms of the amount of sunshine hours. You can see that sunny weather suppresses the biofilm of algae. And if we look at limpet grazing, measured by these wax discs that I introduced you to earlier, you can also see that the grazing pressure is higher in the summer. And that's because limpets are cold-blooded animals, their metabolism's higher in the summer, and they tend to grow more rapidly. But they do get inhibited by very, very hot weather, but they get generally warmed up by warmer water. So you get an increase in activity in the spring, a bit of a dip in the midsummer where they get stressed a bit, and then in the autumn, it depending on when the nice weather is, usually on the Isle of Man, it's May and June, and then their activity tails off as you go into the winter. So this pointed to maybe an effect of physical stress, maybe an effect of grazing pressure. And the only way you can understand these interacting factors is correlation doesn't tell you cause and effect. You have to do experiments. And this is Richard Thompson doing an incredibly complicated experiment where he shaded areas. Some of these areas he excluded limpets, some of the areas he didn't exclude limpets. In some of the areas he put in a control with a clear canopy, a perspex canopy on it. He also got many, many strange glances from the locals by going out on the seashore with a watering can and watering the rocks with nutrients to see if there was any bottom-up effect of fertilization on the algal growth. And what he found was that the only significant effects were shading and grazing. Adding extra nutrients didn't make much difference, even though nutrients are less in the sea in the summer. So by doing this experiment, we were able to unravel some quite complicated stuff, which is probably why we managed to get it published in a decent journal like Ecology. But you've got to have a bullshit model to get your stuff published in top journals. And this is the classical model of top-down control, predators, consumers, intermediate consumers, grazers onto prime producers, and bottom-up forcing by light and nutrients, which applies in many terrestrial and marine communities, particularly in temperate areas. But what Richard and I put in was that actually it's very much modified by what we call lateral modifiers, both in time, season, in temperate areas, insolation stress, desiccation stress, and also over space in terms of where you are on the wave action gradient and where you are on the vertical gradient. So whether something's top down or bottom up can vary with season. I think microbial films are very bottom up in the late winter, early spring, when there's lots of nutrients and there's enough light for them to grow. But as soon as it gets warm, top down control by grazing starts to take hold, but also reinforced by insolation stress. So these are just a few examples of why some rocky shores, and this is one in Hong Kong, end up covered with lots of ironmongery. And this is a very, very dense experiment. There's not a lot of rocky shore in Hong Kong, so they've got to use what they've got very, very strategically, which one of my former colleagues from the Isle of Man did in Hong Kong on grazing with and without shade, looking at the effects on tropical algae, reported in the Plant Animal Interactions book. So if anyone's really interested, these are some of the old classic papers. You could say it's my greatest hits catalogue from the 80s and 90s and early 2000s. That can be distributed to anyone who's interested. So I'm going to do my first pause here. Okay, so I'm going to wave my Devon flag now. This is work that really started when I did a postdoc at the MBA in Plymouth in 1980. And I was strongly influenced by my host, Alan Southwood, who was ex-Port Erin, but then had gone down to Plymouth and worked at the MBA. And he got me interested in broad scale and long-term change. And when I got a job in Manchester, which was not very conveniently near the seashore, I decided that I would continue with this work because it didn't matter if I was in Manchester because I'd have to travel to do it anyway. This is work that I've done over the years since 1980, a lot of it with Alan and Eve Southwood. Eve Southwood's still alive at 90, and we do a shopping for her every week and take it into her. And she's still active. I think she published her last paper in JMBA last year. Sadly, Alan passed away a few years back, but he was a great influence. Okay, so, and this is work done with lots of other people, particularly Mike Burrows and Nova Mishkuska, who's half at Liverpool, half at the MBA. Okay, so the Marine Biological Association of the UK, and I would urge anyone who's got an interest in marine biology to become a member. They've got good cheap rates for students, people from Petroc who are enlisting. There's lots of resources online from the MBA. And they've also got a fab library when it's open. So it's well worth it. And that's where I'm physically based now, except I'm actually in my spare bedroom in Newton Ferris where we've just moved. But they very kindly give me an office. The MBA's got a long history of long-term observing, and I can give a talk on another occasion about the work on fish and plankton and stuff if you're interested. But I'm going to talk about intertidal ecology. And the reason I'm going to talk about intertidal ecology is because it's cheap. And the various time series the MBA have had have been intermittent, but the intertidal stuff has generally been kept going because it's a lot cheaper than taking out expensive research vessels to sea. Though when I was director of the MBA, I managed to restart a lot of the time series. And they're at threat again now, obviously financial crises, other things. So the MBA has both offshore time series, but also the ones I'm going to talk about are ones that Alan and Eve Southwood started in the 1950s. And I've added a few of these. I do Swanage, I do Woolacombe, I do Lynmouth, which they used to do intermittently. And these are ones they visited every year from the 1950s. And why is long-term research interesting now? Well, a lot of the MBA's work was stopped in the 1980s when, during the Thatcher era, it was deemed as merely monitoring. It wasn't testing any hypotheses. But in fact, Alan always had a hypothesis, which was that basically climate fluctuations were driving the change. And I like to show this slide, which is from an early IPCC, because it does show that there was a period of warming, and then there was a cool period followed by a warm period. And for the Northern Hemisphere, it's particularly pronounced. And some of us, in fact, many of the people given the demographic of this audience, remember what a notoriously cool period the 60s, 70s, and early 80s were. Think of all that synthesizer music. Think of flared trousers, long hair. So there was a dip. There was a rise and a dip, and then there's this recent rapid rise due to probably anthropogenic climate change. But probably this secular dip here probably obscured the particularly the North Atlantic region, probably obscured the influence of climate change. And it was a shame that all these time series of the MBA were stopped in about 87, because that was actually the change point. So most people would accept, and this is from the IPC 2014 summary, most people would accept that human activities since 1750 have caused warming due to greenhouse gases. And the evidence has become even stronger since this policy report. I reviewed the 2014 IPCC, so I got a free copy. But there's an alternative hypothesis. There's nothing wrong with alternative hypotheses. This was published by the Singapore Times shortly after Donald Trump became president. And Singapore is a low-lying country facing sea level rise, is very concerned about climate change. This was in the Straits Times. I like this hypothesis. This one particularly offends me. It really does offend me. That's because the grammar is so bad. Even on Twitter, you could at least be grammatically correct. I do wonder actually whether a golden eagle is a bald eagle with a comb over. But fortunately, this particular person has been subject to the ultimate peer review and is no longer president. And Joe Biden's going to take the USA back into the Paris Agreement, which is really, really important. Now, this is a slide I actually presented at a fringe meeting of the 2015 Paris Summit, where I was part of a session sponsored led by the International Union for Conservation in Nature, sponsored by an insurance company. And you know climate change is real when insurance companies take an interest in it. And this shows temperature changes off Plymouth since instrumental records began. It's come off the Hadley Centre website and was done by various of my PhD students that can do this kind of stuff. And you can see since the MBA, the data before the MBA lab was founded was probably collected by the Admiralty, and a lot of this data was subsequently collected by the MBA in Plymouth Marine Laboratory. And there was quite a warm period at the time the MBA was set up, then a cool period, then a warm period, and then there was a very cold period either side of the First World War. And then from the 1920s onwards, there was a warming up to 1959, which is a very, very hot summer. Alan and Eve Southwood got ripe figs in their garden in Plymouth in 1959, and it was the year my brother was born, and my mum had a very hot and sticky pregnancy, but I remember that summer in Weymouth very well. I can also remember the first time I saw snow in 62, 63 in Weymouth when I was 6. And that really heralded in a cold period. There was like a flip, and throughout the 60s and 70s, there were the occasional hot summers like 70, 75, 76, but it was generally a pretty cool period. And when I started working on climate change, it was at the end of the cold period. And I'll come on to that in a minute. But that's my patch of sea off Plymouth. But what is interesting, if you look at the extreme right, and I only update this figure every 5 years, it's due to be updated now, you can see that there was a bit of a dip from about the mid-2005s onwards. And there was a really cold winter in 2009, 2010, and there was an even colder winter in 2010, 2011. And since then, it's got a bit warmer again, but I think peak warm in recent years was probably the late 90s, early 2000s. And again, I'll come on to that later. But I still think it's going up. There's lots of other stuff causing these wobbles, and I'm sure that the global warming's for real, but there's just a lot of weather and a lot of noise in this graph. Now, why are the British Isles and Ireland really sensitive? And this is an old picture by Edward Forbes, who was a Manx marine biologist. And shortly before his death, he drew this map, which was published after his death in A.K. Johnson's Physical Atlas. And looking at museum specimens, he came to the conclusion that to the south and west of Britain, they're what he called southern types, southern softies. And to the north and east of Britain, there were a lot of dead hard northerners who probably only wear a t-shirt in the big market in Newcastle in February. So we got a collision between warm water clades, probably originating in the mid and South Atlantic, and cold water clades. And we subsequently know from molecular genetics that most of the cold water species actually colonized the Northeast Atlantic from the North Pacific about 3.8 million years ago. So all the brown seaweeds, the fucoids, all the littorinids, dog whelks, what else? The kelps, all those major conspicuous elements of inshore communities have got immediate cousins which are North Pacific. And in fact, Semibalanus balanoides, a common barnacle on British rocky shores, also occurs in Alaska, it also occurs in Siberia, and it also occurs in the top end of the Japanese archipelago. And it's not that genetically different. So there's been a lot of interchange, and there's this collision of clades in the British Isles. And that actually makes the region really sensitive to climate change. It's also great as a scientist because it gives you multiple range limits around the complex coastline. If you're working on climate change in Chile or climate change in California, you've just got n equals 1, which is no degrees of freedom with your range edges, or at least here we've got 2 or 3 range edges around the complex British coastline. As things have spread from the south and west into the colder Irish Sea, around the north of Scotland into the colder North Sea, and up the English Channel from the warm Western English Channel to the colder English Channel, Eastern English Channel. So we're fortunate that a lot of work was done by a French guy, Fischer-Piet, and taken forward by Crisp and Alan Southwood in the 1950s. And we've even got some time series of barnacles, Alan Southwood's time series from 1950s to 1987, which I restarted in 97 when I got a job in Southampton, and my limpet time series, which is intermittent from 1980, in fact, to 2020. I haven't updated this slide recently. And we've also got, in terms of the Torrey Canyon oil spill that I come to the end, we've got some shores which were impacted by the Torrey Canyon oil spill and some shores which weren't impacted by the Torrey Canyon oil spill. So Mark Lim did a lot of archive research, got a lot of data together, resurveyed things. To get stuff published, we tested some hypotheses, and Mike Burrows is really clever, and he did some modelling with climate change scenarios. I'm just going to talk really about the resurvey data today. And we had a huge background. These are all the sites for which we had data from Crisp and Southwood, and there was also data from Southwood all the way around Ireland. This is from a report to the UK government, so we left the Irish stuff off. But I managed to wangle some funding for colleagues in Ireland, so we were able to do work in Ireland, and the British team from Plymouth and the Irish team cross-calibrated in Northern Ireland, which might be a bit tricky at the moment with border controls. So this story might have died in the 1980s, but then there was a rekindling of interest in climate change from the late 1980s, 1990s onwards, and I managed to persuade various conservation agencies to fund this work, the so-called Mark Lim project. Now, I got interested in climate fluctuations when I went to Plymouth to do competition between these two limpet species, and I'd read in the literature that they're about 50-50 in the region. I thought, oh, great for experiments on competition. So when I went out on the seashore and found that Patella depressa was much rarer than it had been, I was a bit disappointed, but this prompted a resurvey. And basically the warm water limpet in 1980 and in the early 80s, which was quite a cold period, was much rarer than it had been in the balmer 1950s. And the northern species was more, Patella vulgata was more relatively common. And this sort of sparked my interest in climate fluctuations. I never published this paper because around about 1987-88, I realised that Patella depressa was getting a lot warmer, and the same things were happening with the barnacles. And we delayed publication. And we might eventually publish that paper this year with Mauricio's intervention. Now, Alan Southwood studied warm water barnacles and cold water barnacles. You've got to be a real barnacle nerd to know that that's a Cathalamus. In fact, I put a red dot on it with a marker pen, helps. I think in the top right there is a Semibalanus balanoides. And Alan counted them for many years, and during the warm 1950s, the southern Cathalamus stellatus dominated, now been split into 2 species. And then in the 1960s, the cold water Semibalanus became more common and the Cathalamus decreased. And then when it got cold again, when it got warm again, just at the end of Alan's time series, the Cathalamus were beginning to recover, and Semibalanus was beginning to was doing okay. And then there was a gap, and I resumed the work in 97. And I found that, and I used the same methods and the same shores as Alan, and I found that the warm water species was even more common than it had been in the peak years of the late 1950s. But interestingly, the cold water species is hung in there. And this is one site which is actually 500 metres from where I'm sitting and speaking to you, at the mouth of the Yam Estuary near Newton Ferries in Plymouth, which we were able to keep going when funding ceased for Alan and he was forced to take early retirement. And you can see very nicely the warm water cathalamids doing well in the 1950s, semi-balanus doing well in the 1960s, 70s and 80s, and then the warm water species coming back and accelerating from the 1980s onwards. And also an invasive, Elminius as it was called then, now called Ostrominius, an Australian barnacle that came in on convoys during the war, which established itself in the estuary and is persisting at a low level. As part of the work for Marklim, we resurveyed range edges, and this is Gibbula umbilicalis, now called Steronthophella. When you get to my edge, you could only cope with 2 or 3 taxonomic changes, and then only with ones that you've been involved in. But what used to be called Gibbula umbilicalis in old money used to stop here. Nova found it extended, its range had extended all the way along the north coast. And recently I found some in Orkney, so it actually jumped over to Orkney. So a range extension, this was in the 2000s. And in the English Channel, most things used to stop on the Isle of Wight. A few things would stop, like what was then called Ocelinus, was previously Monodonta. A few things used to stop at Lyme Regis with the odd individual at Portland. But most species used to stop somewhere between Portland and the Isle of Wight. And when we resumed surveys in 2004, myself with Paolo Muscello and with Roger Herbert and with Nova, we found various range extensions of these southern species and molluscs along the coast, beyond their former boundary on the Isle of Wight, which had been a boundary in the 1950s when it was warm, and a boundary in the 1980s when it was cooler. You might ask why these range extensions have occurred on the south coast. Well, I think these warm water species were probably breeding longer and more successfully. So there was a lot more larvae being released into the plankton, allowing increasing the likelihood of spread. But the likelihood of spread was also increased by the proliferation of sea defences which had occurred on the south coast. Actually, a human response to rising sea levels and stormier seas, itself a symptom of climate change. And these artificial structures proliferating all along the south coast probably provided stepping stones. And in fact, we first found Gibbula umbilicalis on these structures in about 2003, 2004. And I recently found Monodonta lineata, Ocelinus lineatus, Phorcus lineatus is the same species, the big top shell here in 2020 and 2019. So these are useful stepping stones for species, artificial habitat for rocky shore species. So at the end of the Marklim project, we found quite a few range extensions of various southern species and also increases in abundance, with Melarafi neratoides, Littorina neratoides, winning as it is spread all the way around British coasts, all the way around. It used to stop here, all the way. I even found it in East Anglia on artificial sea defences. So this was the biggest spreader, and it's got a long larval life. It reaches Scandinavia. Interestingly, we found fewer retreats of northern species. The tortoiseshell limpet, Tectura textudinalis, which used to be Acmea textudinalis, used to occur on the Isle of Man when I was doing my PhD in the 1970s. I haven't seen one since the late 1980s. It used to occur in Anglesey, it doesn't occur there anymore. It used to occur as far south as Bray in Ireland, and you get a few in Northern Ireland. And similarly, it's recorded from Victorian times as going up and down the northeast coast of England. This one is retreated. You get there's occasional odd records from the North Devon coast, and this is a patch of cold water, I think, maintained by the strong tidal mixing of the Bristol Channel. But it's very easy to confuse with Apnea virginia. And Semibalanus balanoides was getting much rarer, and actually in Spain and Portugal, its southern range limit has retracted quite considerably. So lots of advances of southern species, less retractions in northern species. Returning to my limpets, in the early 1980s, the solid lines, they were much rarer than they have been in the 50s, but they're now 60–80% of the population in North Cornwall. They're more than 50% in the South Devon. I could have done my postdoc now very easily that I was meant to do in 1980, and actually Mauricio took advantage of that for his PhD. And also breeding populations were re-established south of the Leen Peninsula. But interestingly, it still hasn't really got around the Leen Peninsula in any large quantities. This is a major barrier to a species that doesn't disperse very well. And Patella depressa never made it to Ireland after the last ice age. A lot of other species did. So this is a poor disperser. Now, this is work that I used to do when I used to visit my mum for a birthday in July every year in Braunton. And when I got fed up with the family, I said, well, I have to go out sampling. And I used to do places I could do within an hour's drive of Braunton. So I've got data from Woolacombe, I've got data from Duckpool in North Cornwall, and data from Hartland Quay. And this is a proportion of the warm water species, Patella depressa, out of the total limpets on the mid-shore. And generally, you can see since I started in the 1980s, it's got more common. There's lots of fluctuations, probably sample variation, warm and cold years, but this is going up. I actually did get around to publishing this in 2008. And Mauricio spent a dusty few days going through my old notebooks and databases, and this is some more recent data, an update to 2015. And there's some quite interesting changes on the South Devon coast. You can see that it's done, Patella depressa has done much, much better at various places. And its range edge in Dorset, it's become more common, again with some fluctuations, possibly a slight downturn in recent years. And on the North Devon and North Cornish coast, these are core sites at Bude and Duckpool. It increased, maybe a slight downturn in recent years. Hartland Quay, an increase and maybe a slight downturn. And interestingly, in Lynmouth and Woolacombe, when I started, you really struggled to find them at Lynmouth, and then they became more common. And at Woolacombe, they weren't so common in the 1980s, and they're now a reasonable population. There's something going on the North Devon coast. So returning to Lundy, Keith Hiscock, who's a great mate, invited me for the first time I met him in 1980 to go do an expedition on Lundy, which is the first time I've been back since my school field trip. And when I was there, I realised that common molluscs, things like Monodonta, Ocelinus, Phorcus lineatus, the top shell, were very rare there compared to pretty common populations on the mainland. The winkle, the edible winkle, Litorhinolitteria, were very rare there. I didn't find one, I don't think, in the week I was there. And the proportion of Patella depressa were quite a bit less than they were on the adjacent North Cornish coast. So clearly some things were not reaching Lundy, probably because it's an isolated island, that these have got larval dispersal. Any individuals probably got there rarely, and probably because of dispersal around Lundy, very few of the larvae would actually come back. They probably get lost. Gibberellum bilocalis was in most places, but nothing like the numbers on the mainland. The other thing is there's obviously a bit of a break going on for Patella depressa here. Pretty common up to Hartland Quay, and then as you got into Barnstable Bay, and I'm a Barnstable boy, so it's Barnstable Bay, it was getting much, much less. And if we go back to Mauricio's work, we can actually see that this boundary around about Hartland Key, if anything's been slightly breached, and it's sort of made a small leap, and it's doing pretty well at Woolacombe now. Now, I'm still doing this work. This was 7 days before lockdown with actually one of my students and another one of my students that I'm co-supervising, visiting the shore that Alan Southwood and Eve Southwood first took me to in spring 1980, just 7 days before lockdown, before a dodgy night out in Weymouth, the last dodgy night out I had this year. And if we look at some data that Mike Burrows gave me from Scotland, and the same kind of things going off in Plymouth, but Mike had updated the data recently, you can see that there was this rapid warming, but actually things have just cooled down a little bit in recent years, particularly in Cornwall, particularly in northern Scotland and Ireland, but they have also around us as well. And Mike's very clever. He's probably the cleverest of my PhD students. I didn't realise it at the time because he was my first, but he's got a very complicated index where he looks at the geographic affinities of different species and works out a community index of the 60+ Markland species that we look at using semi-quantitative abundance scales. And we started in 2005, and this is the data for Southwest Britain. So as a whole assemblage, things are actually tracking the perhaps slight downturn in temperature, particularly this cold spike that occurred in sea surface temperature in the late 2010, and this is the same pattern off Shetland, slight downturn, and perhaps the animals being a bit more sensitive than the plants. And remember, many of the plants are actually got northern affinities. Now, if we go to the English Channel, though, we're still finding range extensions, even though it's got a bit cooler. I was really excited this year. I got permission from DEFRA to break lockdown to do sampling. Bloody difficult parking in National Trust car parks, but we managed to do quite a bit of work. And I was very excited to find breeding populations at Bognor Regis of Ocelinus lineatus, now Focus. I'm an author on that paper, so I feel slightly embarrassed about that name change. And what happened there was the sea defences, which I showed you earlier, had actually modified the substrate behind them. There was a boulder field, and because of lockdown, I didn't park in the usual place I parked in because it was closed, because the pub was closed. I had to walk along the beach, and I walked across a different patch, and there was a boulder field that had been created in block number 3. And I usually just go straight to block number 5 from the west. And I actually found this breeding population, and they were quite old. They'd been there for a few years. Just because where I parked my car meant I hadn't spotted them. And similarly with other species. And even with Patella depressa, we found odd individuals getting as far as Beachy Head. So things are still spreading. And I think that's because once they've breached this biogeographic boundary, this hydrographic barrier, a bit like Hartland Point, a bit like the Fleam Peninsula in North Wales, they've been able to establish populations, and these populations have spread. Gibula umbilicalis, which I think has quite a short planktonic larval distribution, maybe only 2 or 3 days in the plankton, is a bit like the infantry in warfare. It can make a little spread and consolidate, and this is the best spreader, and it's all the way along the English Channel coast now, and got breeding populations right up to Beachy Head, whereas previously there used to be just a few individuals on the Isle of Wight in the 1980s. So let's have a look at the clock. Just to link ecology with climate change and pollution, I'm just going to very much talk about the Torrey County North spill. This happened when I was 11. I can remember the hunter bombers taking off from Chivenor to go and try to bomb the Torrey Canyon with napalm to burn the oil. It didn't work. And this could be viewed as a very inadvertent large-scale experiment. And it also illustrates some of the interactions of climate fluctuations and other pollutants like TBT during recolonisation. So a big disaster. acute pollution, but made much, much worse. On 10,000 tonnes of dispersant, 14,000 tonnes, on 14,000 tonnes of oil, 10,000 tonnes of highly toxic, first generation organic oil slick cleaners were applied. Basically an organic solvent with some palm oil to stabilise it in a surfactant. Absolutely perfect recipe to rupture cell walls. So there was massive sequences of colonisation following the kills of limpets. This was early work done by Alan and Eve Southwood. And the reason I heard him give a talk and the reason I chose to do a postdoc with him in Plymouth, and that's where the Torrey Canyon went ashore on autopilot. The crew at breakfast, they forgot the Isles of Scilly were in the way. Alan Southwood had all these stations in Devon and Cornwall. So great control areas, but also impacted areas from the oil spill, from Travone in the north round to Lizard in the south. And following the death of grazers, there was a massive proliferation of seaweeds on the seashore. This was previously a barnacle-dominated seashore, completely covered by fucoids on the low shore and green algae on the high shore because all the winkles got zapped as well. And these are pictures that Eve Southwood collated for me a couple of years back for a conference in California on the 50th, where I talked about the 50th anniversary of the oil spill. And you can see whole shore covered in seaweeds for the first few years, and then getting bare again. And there was a very bare phase in the mid-70s, and then getting back. Alan Southwood and I reckon it was getting back to normal sometime between 81 and 84. I first visited in 1980 with Alan, and we decided not to stop the time series and keep it going. And this is probably normal business is consumed. This is a pretty exposed shore. You get the odd patches of seaweed from time to time, but primarily barnacle dominated. But occasional outbreaks of racks of fucoid seaweeds. So that's the seaweed dominated phase, that's the bare phase, and that's some data. Greens, fucus, just like the exclusion and removal experiments, a bare phase because of all these limpets. These limpets then starved and died. Because they died, there was a second burst of seaweeds. And if I can grab a ruler. It's a bit like a damped oscillation with an overshoot and things coming back to normal and then just getting to a normal level of oscillation, reflecting natural fluctuations and recruitment of the key species. But some species were very slow to return, Patella depressa. And if you wanted to, you could argue that they hadn't, that no recovery was very slow in occurring because this species hadn't recovered. But this was due to climate, not due to the oil spill. We know that in the 1980s, this species was very rare everywhere in the southwest of England, or much rarer than it had been in the 50s. So there weren't very good source populations for it to come back. We also know from some work that another student of mine has done that Patella depressa does not like being underneath dense fucus. And we know from Mauricio's experiments that Patella vulgata beats up Patella depressa under dense fucus. So this is the time series, this is fucus, this is the oscillations, this is a natural recruitment event that occurred in 2000. And then the fucus goes down again because limpets aggregate around it, munch it down, and then there's another escape. And then just to celebrate the 50th anniversary, there was a really big fucus escape in 2017. And this is the limpet data, the cold water Patella vulgata coming in, and fucus is a really good nursery ground for Patella vulgata, not for Patella depressa. So a massive population explosion causing the decline of the fucus, then a crash, and then Patella depressa coming in, and Patella depressa doing well during bare periods, and Patella vulgata doing well during fucusy periods, because it aggregates under fucus, and the juveniles do well under fucus. And then coming back to some kind of normal, and now the warm water species winning in Mounts Bay over the cold water species. But there's one hermit crab, Clibonarius erythropus, which was first discovered by a schoolboy, an amateur, in 1959 at Mosul. And even Alan Southwood studied this for many years. And we've been waiting for this to come back, and it never came back. It also used to occur at Wembury in Devon. This got named St. Piran's crab after a pole in Springwatch, because it was Cornish, but it's always lived in Devon. It's always been in Wembury. Why didn't it come back? Well, even Alan Southwood suggested that one reason it didn't come back when it got warm again was because of the lack of dog whelk shells, because dog whelks got really badly affected by tributyl tin pollution, with the female dog whelks growing a big penis, the reproductive glands getting blocked, and the populations getting wiped out as the females got killed as their blocked reproductive ducts ruptured, and it killed the animal. So dog whelks became very rare, as did other whelks, which were similarly affected. This is Nazarius hinea, not sure what it's called these days. So this is data from Marazion, where we took Eve Southwood to have a look for it on many occasions before she became a bit infirm to go on the shore. We never found it. It got zapped by Torrey Canyon oil spill. It recovered a bit, but it eventually pegged out. But this had nothing to do with the oil spill, because exactly the same thing was happening at Wembury, where the population, which had probably recruited a couple of times from France in the warm water period, eventually died out. So, but why didn't it recover when it got warm again? Maybe lack of dog whelk shells. So when you're looking at pollution, you've got to put it into a much broader context. You've got to look at overall climate change, and this is the window, getting colder and then getting warmer again, and also the Torrey Canyon, which would be called a pulse disturbance, or a chronic pollution event, an acute pollution event, or TBT, a more chronic press disturbance, which persisted for a long time. So there's a lot of short-term and longer-term impacts at local and more regional scales superimposed on pollution. And you need to have this long-term broad-scale data to be able to understand what was going on. And these warm water species only recovered when it got warm again due to climate fluctuations around about here. We also know they don't do very well under seaweeds from experimental ecology. And you can also get considerable time lags. It took a long time for Clibonarius to get over from France. But the day that I should have been giving the talk in November, I did go out on the shore and I found some very, very good populations in Cornwall. And I also managed to get out in October, and there's some very good populations east of Wembury now. So they're spreading. They're as far down as Bigbury on the South Devon coast. So that's hot off the press. That's data from this year, collected during lockdown. So my take-home messages is you've got to look at all impacts: climate change, fishing, non-natives, habitat loss is particularly important. Rocky shores are great. They're easy and simple to sample. There's good ID guides, even if the names are probably need updating. Lots of historical information, lots of background knowledge from experiments so we can understand the mechanisms driving change. You can use rapid assessment methodology. You can do really complicated stats, even on semi-quantitative abundance scales, which is what Mike did with the paper we published this year. You can survey them easily. You can separate out global change and local changes due to pollution, red tides, reclamation. You can possibly show whether MPAs are working, and if things change in an MPA, you can diagnose the right causes of those changes. So without this long-term and broad-scale pattern data within parallel experiments, and also modelling, which I haven't talked about today, to enable prediction, you can get some idea about what goes on, and you can avoid erroneous interpretations of responses to climate change, and also impacts and recovery from impacts. And there's great potential after an initial professional phase to translate into citizen science. And there was the COCOS project, which Nova Mashkushka and Mike Burrows were involved in. I was dean at the time, and they thought I'd gone far too far down the dark side of university administration to take part in such things. But there's a limited suite of easily identified species. You can use the power of the smartphone camera to get to get authentication of your records. But I still think you need annual professional surveys. Not everyone can identify limpets and barnacles properly in the field non-destructively. No, sadly, my wife Elspeth can, but despite working in the pharmaceutical industry, she's been dragged down many rocky shores between March and October, and sometimes she complains. But I do say to her, at least I've given up cricket now. You're not watching me play cricket at weekends. We're going to nice seashores and counting limpets and barnacles rather than watching a game of cricket. So thanks very much for your attention. And we've still got a bit of time for a few more questions, but I'm very happy for anyone to send me emails. It's sjhawkins@sotton.ac.uk. Not my MBA email address, I never look at it, to answer any questions. Okay, I'll stop now.